So I'm going to talk about variant identification, and let's think about the overall process here. So we've talked about having two sequences, aligning them together. Then what we've talked about is, how do we do this on a bigger and bigger scale with many sequences? The gymnastics we have to go through. We've talked about making patterns of sequences and maybe putting a lot of sequences together. Where we're sort of going is, kind of the machinery of dealing with personal genomes. And one of the things we want to do in that context a lot of times is take a sequence, align it to the personal genome, and then find where it's different. So that's looking for what's called variants in the sequence. And so there's a lot of, and actually in the context of personal genomics, there's a kind of you're looking for a pattern, but it's a very specialized pattern where you're going to align a bunch of sequences, and now you're going to look for columns in that alignment, in a multiple alignment, that are together different, and those are usually SNPs or potentially indels. And so we're going to talk about those a little bit, and then talk about a somewhat more complicated variant called the structural variant that arises. Okay, so let's do that. So now I'll kind of sort of go through that graphically here. So the main processes we have now in resequencing a genome, personal genomics, what happens when if you have your sequence done, is you'll generate the reads from the sequencing, and they're sort of diagrammed here, and then you'll map the reads to the genome. Okay, so each of these things is the genome is the database, the reads are the queries. You're doing a vast amount of database lookup and so forth, and then you're essentially doing a little bitty multi-alignment, and in any given window you have a whole bunch of things matched up, and then you have the same idea if you have kind of like conserved columns and so forth. Now, in this case, almost every column is conserved. It's kind of different from like the cross-species thing, and you just have particular columns that have variants. And so when we have those variants, we're going to call SNPs if we have a lot of differences in the reads relative to the target genome or the reference genome. We're going to say there's a difference, and we'll maybe call it a variant. Now, then there's also the notion, which is a little bit more complicated, of the genome that we have has maybe a duplication, a deletion, an insertion, some complicated thing going on relative to the reference. And when we map the reads back, we're going to get a very funky pattern. Maybe a lot of reads are going to map in the duplicated region. Maybe they won't map where there's deletion and so forth. And so we'll call here what's called structural variants, which are essentially much more complicated and larger variants to call. And then obviously there's a potential, for instance, diagrammed at this spot, for the new sequence to actually have a new inserted sequence. And so we might even have to do a small amount of assembly for assembling these new sequences. And we don't really go through assembly in this class too much, but assembly is a big topic in bioinformatics where people talk about how you assemble sequences. And then in addition, for the variants that we find here, the SNPs and the structural variants, there's a process called phasing. And what we want to do here is, because actually the human genome that we have is fundamentally diploid in nature, it's not haploid. For any given variant, or any pair of variants, we have to decide, are the variants together on say the maternal copy, or are they one variant on the maternal, one variant is on the paternal copy? So we have to phase the variants together. So we have to decide, for instance, if we found these two variants, are they on the same same haplotype in the diploid, or are they different haplotypes? And then we want to do that over a long block where we get all the variants kind of phased correctly. And that's the process of phasing. So now I'm going to just tell you a little bit about the SNPs calling, just to kind of fill it up. It's a very important process. I'm not going to go into it in detail, but I just want you to get some notion of how this is done. Now, before I also say that, I just want to make clear too that there's two types of variants that people tend to find for, say, single nucleotide variants. There's germline variants, and those are obviously come from sequencing, say, our blood and so forth. And then there's also somatic variants. Somatic variants are things that are not in our germline. They're developed in our non-germline tissue, our soma. And these typically occur in a tumor. Now, you can, of course, get somatic variants in any cell you have, but most of them you tend to get in a tumor. And these variants are a little bit, just everything's a little bit different. There's, the notion of the diploid genome and phasing and things like that are a little bit different for the somatic variant relative to the germline variant. And then also the somatic variant, there's, I think, a lot of sensitivity you have to think about contamination with germline variants and, how much germline you have, how much somatic you have in a given sample. Okay, so how do we find single nucleotide variants from a big stack? So you imagine now what we do is we do a massive multiple alignment. We stack up all of these things using whatever BWA or whatever. And we want to know if this column, which seems to have some aberrant bases, if it corresponds to a variant. And the simple idea here is if we observed, if we had truly perfect sequencing and we believed every single read we got, we might say, even if you have one read that has a different thing, we would believe that. But pretty much we don't believe that. What we want to do is we want to observe an appreciable number of variants relative to what we expect. And if we are expecting a heterozygous, for instance, if we're expecting the person has a germline heterozygous variant, okay, then we're kind of expecting that if the reads are sampled, kind of correctly, we're roughly expecting half of those reads would have the variant and half would not. So we'd get something that looks like this. But now that's just a statistical expectation. We wouldn't see 50-50 all the time. It could be, at one spot in the genome, we would only get, if we had, 10 reads, we'd only get maybe 3 with the variant. And that might be a reasonable thing. And we could kind of call that as a variant, just kind of depending on how many we got. We might not believe it, for instance, if we only got 1. So people often develop the sort of statistics for this in terms of Bayes' theorem, and I'm going to describe this to you. So we want to figure out what's the probability of getting a particular genotype given our data, okay? And what we basically, and the data is what we observe here. And so we're basically going to invert that and talk about the chance of a given genotype, that's having a heterozygous SNP, for instance, to give rise to this data, okay? And then, of course, we have the probability of that genotype and the probability of the data, okay, and so forth. And that's the sort of standard inversion that we do. And now, if we kind of think about how we might think about this in a very simple kind of error-free type of situation, we might imagine that if we have a heterozygous SNP, its probability of generating, the alternate allele is a half, okay? And so, we would get a simple kind of binomial estimate of how often we would see, 5 out of 10, 4 out of 10, and so forth. And this would essentially give us our probability of, having a heterozygous SNP at that position, or the probability, I guess, of having a homozygous SNP as well. Now, of course, this particular probability of having the data given the genotype, we often want to modify this probability to have some sense of how likely we would get this in a kind of not an error-free context. So there's a whole complicated discussion of how you can model the sequencing error and so forth. We're not going to talk about that. I just want to give you a quick sense of what we might get. So that's the finding SNPs and SNVs, just simple binomial model and so forth. So now we're going to talk about structural variants. That's this SVs. And I'm going to spend a bit more time on this because this is a much more of an active area of research. And lots of people have ideas for how you find structural variants, and it's very complicated. And this is more of an open-ended research area. So there's a number of basic categories of methods that people have for finding these things. There's one method is kind of what's called paired ends, where you look at if the sequences are sequenced in a paired end fashion, you look at how the paired end separates, and that maybe helps you find a deletion. There's also the notion of a split read, where if you imagined you allowed the read to align and the read actually aligned in a split way, you would call that split read. There's another method called read depth, where you basically count the read stack at any point. And if you saw there was, for instance, a big drop, you would say there's a deletion, and then of course local reassembly. And now I'm going to go through these different methods. The simplest is read depth. And originally this method was actually developed pre-genomic sequencing. People were just looking at the like what's called an array, a hybridization array for oligonucleotides. And then these methods were readily adapted to next-gen sequencing. So I'm going to kind of describe kind of a classic way this is done and then explain how it gets adapted. So basically what you imagine happening here is you have this is your individual's genome, right? And you have, the person has a deletion here, okay? And now these are the reads you're getting from that individual. And you're going to map them all to the reference genome. You're going to map them. And then what's going to happen, because you only have half as much genome here where you've cut away the deletion, the expectation is that if you just looked at the depth of the pileup at any given spot, you would get a signal that looks like this. It would kind of drop where there's this deletion. And this is a picture of like a real signal. These are very noisy signals. And as I'll describe to you, there's a lot of processing involved. And this is actually an example of a signal here. You can see it's very, very noisy. And it has to do with the fact that sequences map, there's a lot of potential of mismapping, and it's a very statistical sort of quality to this. So it won't look quite perfect. And what people spend a lot of time doing is how they find a kind of sort of simplified model of this, where there's a clean deletion here from this noisy signal. And this, of course, would show maybe a duplication. But then again, is it a simple duplication, or is there a kind of cutaway version right here? It's hard to look at these signals. And so now I just want to show you one way of doing this. And this is based on a hidden Markov model. And I picked this approach because I thought it just nicely gels a little bit with what we talked about in just talked about for multiple sequence alignment. So you're given a sort of very noisy signal profile that looks like this, okay? And this is your observed data. And what you want to do is, now you have a model, but now this model doesn't emit sequences, it emits signals. So you have a state here that represents sort of the normal diploid state. You have a state here that maybe represents a lost state where you have a deletion, and a state here in red which represents a gain state. So this state would maybe emit twice as much signal, this state would emit less signal, and this state would emit the amount of signal here, okay? And now what you're going to do, and then you have a sort of a transition probability to transition from these states, to stay in the sort of normal state, or to transition from the normal state to one of these other states. And so you're going to take this sequence of data, which is a bunch of continuous numbers, and you're going to fit it to this model, okay? And you're going to see what's the most likely sequence of hidden states. That's these red, blue, and green things that match. And then the sequence of hidden states tells you, are you in the gain state? Are you in the deletion state? And it gives you a particular thing here. And so this actually just shows a little bit more detail on this model here, where you have your transition states, you have to parameterize all these probabilities here, and you have your match state, and your match state emits different chances of having that particular signal. Now I'm going to show you another type of approach here, which is a somewhat different approach, but it's so what's the problem with that that read depth approach? So you have to have a model for the things. You have to parameterize these match states. How do you parameterize them? Well, you can train them, but there's a whole process here. And a lot of times, maybe you don't want to have any prior assumptions about, how many deletions you have, how big they are, how to parameterize this. So there's another set of models that people have used, or another set of models there are another set of approaches that are not model based. And so, for instance, I'm going to describe a particular model here that's called this mean shift based approach model. And this is very much like almost like an edge detection or boundary detection method in computer vision, where you're just going to look at kind of the density of points, in a given region, and try to figure out if they kind of collect into a particular bin, and then you'll get these kind of change points. It's almost like, as I'll describe to you in a second, it's like you're looking for where rivers flow, and when you pass the continental divide, rivers flow in one direction versus flow in the other direction. That's a kind of change point between, I don't know, the west state and the east state, and that would have, in your genomic terms, that would correspond to a spot between a deletion. Now, this is a little bit more intuitive how these type of algorithms work. Just a little bit of intuition here. So what you do here is and I'm going to show you this, more of an image processing way. On a 2D, it's somehow a little bit easier to see on a 2D thing how you would find a boundary, though the process works very similar in 1D. Basically, what you do is you go to a particular spot in the signal, okay? And then what you do is you find the sort of center point of that. You have sort of kernel here, and then you try to find a local gradient. If you just look at these points here, you can see if you try to sort of estimate the sort of density of points, you'd see maybe it's kind of increasing here. So there's a local gradient going in this direction. So you follow. Then you take your center point and you shift that thing here, and you find another center point, and you re-estimate your gradient at that point. And you keep doing this, and what's going to happen at some point, of course, is your gradient is going to stop being appreciable, and you'll kind of stop at a particular region. And that's a sort of like a basin, okay? And you'll find these particular different kind of basins of attraction, and then you'll find the division points between them, where you sort of start and you go to one basin, and start and you go to another basin, very much like the kind of the sort of river flowing analogy I described, and that will give you boundaries between your, say, deletions. So you'll sort of start off, you'll find out what basin you go into. It's like you're standing up, I'm making up in like the South Dakota, and you track where you're going to go down following the gradient. You walk someplace else, you track where you go down from there, and eventually you'll find that at some point you'll go to two different completely places. That's going to be a boundary in this thing, and at that point you would call a genomic say a boundary at a deletion. So now I'm going to tell you about another method, and this is paired end mapping, and this is a very different type of approach for finding deletions and duplications in the genome. And this method takes into account the fact that often the way sequencing technology is done, people end up sequencing the two ends of a fragment, and they know they have a long fragment, and they sequence two ends. I don't know if people can see what I'm doing here. They sequence two ends, and they know that the ends are proximally separated by the sort of size of the fragment. And so then what they can do is when they map the ends back onto the genome, they can see, do the ends have the same separation because they were once all part of that big fragment, or are they farther apart or closer apart, or even if the orientation of the ends changes. And this is very useful in genomics. It's like I said, it's not so intuitive, but if you do this, you can find, for instance, that the ends are, say, closer together than what you expected. then maybe there's been a deletion. Or if one end is flipped relative to what you think, maybe that you've mapped the things onto a place where there's been a genomic inversion. And there's a variety of statistical ways of looking at how different those are, and those are called paired end mappers. And then I'll just tell you, just to fill this out, there's also split read mappers. And split read mapping is, it's also, we're not going to go into detail on this, but you can sort of see what's happening. Now we have to go back to the mapping process. So everything we've done so far has kind of been based around, we're going to use BWA or BLAT or one of these programs that's doing the hash tables or the Burrows-Wheeler transform, and it's going to map, it's going to take our read and find the best spot it maps. What if our read actually looks like this? It maps, here's our read, part of it maps to one side of the deletion, another part maps to another side. There's no way that one of those mappers is going to find it. So now what we can do, which is fine, is we have to retool a mapper. And so we have to look for a way of mapping split reads. And so this becomes, I won't go into obviously the details, but it becomes very computationally challenging. So we no longer will be able to potentially map things as fast as we can with one of these highly optimized mappers. And so we have to think of how we can find these particular split matches. And people have a variety of different ways of doing this. I mean, usually what they do in these things is they map all the reads, they get all the ones that map easily, then they're left with a few reads that don't map, and they use this more complicated mapping approach to find find where the read might split. And then of course the split doesn't find the entire well, for a deletion it would span the entire thing. For an insertion it might not do that, but it will sort of tell you about the endpoint or the breakpoint. And it's very useful for finding an accurate estimation of the breakpoints. Now, of course, it gets complicated. So if you actually take a split read mapper, here's a deletion, here's its split, that's fine. If you have a small insertion, life is good. You take your split read mapper, you map it here, you map it here, and actually you have the sequence of the insertion because it's in your read. However, if you have a big insertion, things get more complicated because now your read no longer spans the entire insertion. So you will not get a read that will go this entire length here. So the best you can get is something that looks like this now, where one end of the read maps and the other doesn't. And then so now you don't really know the full scale of this insertion. So now it's going to be more complicated. You're going to have to find some other point here and then group them up together. And then it gets even more complicated. If you have a region of the genome that, imagine it duplicates, and maybe it duplicates and then even inverts, you're going to get very complex patterning with split reads. I won't go into detail, but I think you can see can get a little complicated. Now, I just want to show you an application of split read, how people practically do this or how you would encode this in a kind of simple way. And this is, so a lot of these encoders for split reads are very custom. They're built to do a particular type of split read. And there's really not a general approach. So I'm going to describe one approach now for finding what's called retroduplication variation. And basically the idea is that a lot of the variation we have in our genome comes about through what's called retroduplications. And what happens here is you have a gene in the genome. The gene is transcribed. It's spliced. So that means all of the introns are taken out of the gene. And then this spliced product is reinserted in the genome, and that creates a retrogene. Now you might say to yourself, this seems very complicated, but it turns out this is the dominant process, or one of probably the dominant process of genome remodeling for us. And most of the human genome is made up of these retroduplicated elements. You maybe heard of like lines and signs and alus and things. Though these are these types of elements, and it's actually somewhat complicated to find them. And one particular type of thing that these also give rise to is processed pseudogenes. I'm very interested in pseudogenes, so I'm particularly keen on this. And these are where you have a gene, it's transcribed, and that transcribed copy is reinserted back into the genome. So now what happens? What happens now? So now you have one person has this copy reinserted, another person doesn't, and a third person does, okay? And now that's if the retroduplication is known in the, say, the reference genome, but imagine it's not even in the reference genome. We don't have a retroduplication at all in the reference genome at all. The person we're sequencing has a novel retroduplication. Then what we have is the situation looks like this. And this would be a kind of a sort of insertion situation. So now what I want to show you is how do people do the split read mapping. What they do here, which is kind of cool, is they basically say, okay, we don't know how the read's exactly going to split, but there's a clever technique. They know that all of the junctions that you would have in this type of variation come from splicing, right? So you have the exons being spliced together. And so what they do is they make up a huge splice junction library. So they'll take all of the sequences that could have been spliced together, and they'll say, we don't know, if we have a retroduplication or whatnot, but we're just going to make up all possible splices from that. Okay, so they'll take this, and this. They'll imagine that maybe there's a splicing that has this and this, the blue and the green, they'll splice that together. So they'll make up a huge splice junction library. Then what they'll do is they'll take the aligned reads and they'll align the reads to the reference genome, and then they'll align them back to the splice junction library, okay? And they'll find, actually, sometimes they map onto the splice junction library, okay? And so now they essentially, this is a split read alignment. So they'll have properly aligned. Now they don't know, this is a little complicated now, so they don't know where in the genome they are. They just know, here's a splice. I know I aligned to the splice, but I don't know where I am in the genome. So now what happens, and often in paired end sequencing, they'll have another end of the paired end that will align someplace else in some real place in the genome perfectly. So what they'll have is they'll have a split read alignment in one place that aligns to the splice junction library, and another part of that paired end that aligns into the genome. And then they can kind of sort of say, oh, right around that spot, there was probably an inserted sequence. And then there's other techniques they have and so forth. But that's the kind of quick sort of thought on that. So now I'm going to stop for questions on the